We have been talking about endocytosis. I hope you remember when we were discussing immunology, we talked about how professional phagocytes engulf foreign particles or bacteria or pathogens. So let's look at the molecular events of this process in more detail. The material to be ingested is progressively enclosed by a small portion of the plasma membrane, which first invaginates and then pinches off to form an endocytic vesicle containing the ingested substance or the particle. This basically is also called phagocytosis, which involves ingestion of a large particle, for example, a microorganism or dead cells, by vesicles called phagosome. This is to distinguish phagocytosis from pinocytosis, which I will talk about later. But phagosomes are basically larger vesicles which are greater than 250 nanometer in diameter. Pinocytosis involves ingestion of fluid and solutes via small pinocytic vesicles which are about 100 nanometer in diameter. Pinocytosis basically means drinking. So many cells continuously form these vesicles and enclose the extracellular material, fluid, and also by doing that they capture small ions that they can use for their functioning. Phagocytosis, basically eating of the cell. Most eukaryotic cells are continually ingesting fluids and solutes by pinocytosis. Large vesicles are most efficiently ingested by specialized cells, phagocytic cells, that we have talked about when we were discussing immunology. Here is an electron micrograph of showing the process of a phagocyte pinocytosis. You can see the vesicle is being formed. The membrane is invaginating itself. It is folding inside, and once it does that, there are special proteins that basically tighten up at the junctional part of this membrane, and ultimately it is cut off and a vesicle is then released in the cytoplasm. In mammals, there are three classes of white blood cells that act as professional phagocytes. We have talked about them: macrophages, neutrophils, and dendritic cells. We talked about these cells in fair detail when we were talking about immunology. These cells develop from hematopoietic stem cells, and they defend us against infections by ingesting invading microorganisms. Macrophages also scavenge senescent cells and cells that have died by apoptosis. We talked about apoptosis, or programmed cell death, where the remnants of the cell, after the cell has died, they're ingested by these professional phagocytic cells. In quantitative terms, the later function is by far the most important our macrophages or phagocytes perform. Macrophages, for example, phagocytose more than 100 billion senescent red blood cells every day. So every day our phagocytes, macrophages, they eat about 100 billion red blood cells which are not functioning properly. Particles first bind to the surface of the phagocyte. Phagocytes have a variety of special surface receptors that are functionally linked to phagocytic machinery of the cell. Remember, we talked about Fc regions of the antibodies. These Fc regions, or the tail regions of antibodies, are recognized by Fc receptors present on the phagocytic cells on their surface. These receptors bind the Fc domains of the antibodies, which have coated, for example, an bacteria, and with the help of this interaction, Fc region and Fc receptors, these phagocytic cells coat the bacteria with their membrane, ultimately engulfing it. And this is the process of phagocytosis is triggered when the receptor, Fc receptor, binds the Fc region, activating the cellular machinery, which will of course also reorganize cytoskeleton and help the cell ingest the bacteria. So also apoptotic cells, they lose their asymmetric distribution of phospholipids in the plasma membrane. As a consequence, negatively charged phosphatidylserine, which is normally on the cytosolic, which is present on the cytosolic face of the lipid membrane, is actually flipped. It flips, and now it is exposed at the extracellular part of the plasma membrane. This is also recognized by phagocytic cells, and they trigger phagocytosis of the dead cell. Here you can see the process. Here is, for example, a bacteria. Actually, if you notice, this bacteria is in process of dividing, becoming from 1 to 2. And here's the membrane of the phagocytic cell, which has formed a pseudopody or a false foot. And this false foot is going to ultimately encircle the bacteria, and a vesicle will be pinched off, and this vesicle will go in the cytoplasm, where it will fuse with the lysosome, resulting in degradation of this pathogen. Here you can see phagocytic cell is actually in the process of eating 2 red blood cells. These are both electron micrographs. This one is a transmission electron micrograph, and this is the scanning electron micrograph. I would also like to talk about receptor-mediated endocytosis. Basically, this bacteria, engulfing of bacteria, is also receptor-mediated endocytosis. Here's another example. The low-density lipoprotein particle, or LDL, is basically also ingested by cells by receptor-mediated endocytosis. Here on your screen, you can see the LDL particle. It has cholesterol molecules, and it has phospholipid monolayer, cholesterol ester molecule, and a protein molecule which is holding the structure together. Each particle contains a core of about 1500 cholesterol molecules esterified to long fatty acid chains. So if you notice, here is the cholesterol molecule, and this tail part of this molecule is basically the esterification of long chain fatty acids to this cholesterol molecule. So here's what happens. When LDL binds the LDL receptor, it causes a conformational change in the cytoplasmic domain of this receptor, which is recognized by a protein called adaptin. This adaptin, or basically if you want to think of it, it's basically an adapter that will link this receptor to the clathrin molecules, which are actually the molecules that form, which are the coat molecules. So when this interaction takes place, the clathrin coated pit is formed and results in accumulation of receptors bound to LDL particles. Ultimately, this clathrin coated pit will form a spherical structure, which will ultimately result in formation of a vesicle. So here is our LDL receptor bound to LDL. Here is the adaptin molecule. Adaptin is basically serving as an adapter between the receptor and the clathrin coat. So basically, the way these clathrin molecules fit together, just like football patches, they form a spherical structure. So which forces or bends the membrane, and the membrane also attains a spherical structure. Now, how do you pinch off this membrane? There are additional proteins. For example, dynamin is one of those proteins, and other proteins associated with it. They are thread-like proteins, so they form, they encircle the narrow region of this vesicle. And once, if for example, if you take a thread, you run it around a ball of dough, and if you pull on the thread, you will divide the ball of dough into 2. I gave you that example when I was talking about cell division. Similarly, dynamin basically results in completion of the process of vesicle formation, resulting in release of this vesicle. As soon this vesicle is released, of course, it will lose its clathrin coat, and the membrane is now exposed to the cytoplasm. So here's the whole process. We have LDL molecule binding to the receptor. This receptor having a conformational change in its cytoplasmic domain and resulting in formation of association with the clathrin molecules, which result in formation of the spherical structure. As soon this vesicle is released, the clathrin coats will disassemble. Now the membrane of this vesicle and the proteins protruding from it are exposed in the cytoplasm. Here, this vesicle is going to fuse with early endosome, and from early endosome, the receptor for LDL will be recycled by other vesicles and be sent to the plasma membrane. Whereas the LDL particle will end up in the lysosome, where the hydrolytic enzymes will work on the contents of this vesicle and which will result in release of the free cholesterol. So basically, this concludes our discussion of protein targeting and vesicle transport. We will talk about more applied part of the cell biology in our next module.